Hello. In this video, we will learn about transport proteins, which include pumps, channels, and carriers. Let's get started. In the video on biological membrane, we have seen that the membrane is made up of lipids. Lipid-soluble substances like oxygen and carbon dioxide can dissolve in the membrane and therefore can cross through the membrane itself. However, lipid-insoluble substances like sodium and potassium ions cannot dissolve in the membrane and therefore cannot cross it. Some molecules like glucose are simply too large to penetrate the membrane. So for such substances, we have various transport proteins on the membrane. They create a passage in the membrane for the movement of such substances. All the transport proteins are transmembrane. This means they go through and through the membrane. Well, they have to create a through and through passage. There is no other option. There are three main types of transport proteins: pumps, channels, and carriers. Let's see each type one by one. First, pumps. Their specialty is to move substances uphill against their electrochemical gradient. For this, they use energy from ATP. For example, sodium-potassium ATPase pump. Sodium concentration is higher outside and lower inside the cell. But these pumps move the sodium outside the cell using energy from ATP. This type of transport is called primary active transport. So this was about pumps. Now let's see channels. They simply provide continuous passage through the membrane, just like a tunnel. Ions can freely diffuse through this passage. Thus, movement through channels is passive down the electrochemical gradient. And channels are often selective for a particular type of ion. Now, the channels can be of two types: pores and gated channels. Pores are always open. For example, porine, nuclear pore complex, and aquaporine. Gated channels, on the other hand, are not always open. They have gates that control the opening or closing of the channel. When the gate is closed, ions cannot move through it. But when the gate opens, multiple ions can flow through its pores simultaneously. These gates in turn are controlled by ligands or voltage. Respective channels are called ligand-gated channel and voltage-gated channel. Ligand-gated channels open or close depending on binding of certain ligands. For example, sodium channels at the muscle end plate open on binding of acetylcholine. In voltage-gated channels, voltage across the membrane controls opening or closing of the channel. For example, voltage-gated calcium channels in the heart opens when the membrane depolarizes to a certain level. So this was about channels. Now let's talk about carriers. They never provide continuous passage. They have at least two gates and both never open at the same time. They first open at one side, binds with a molecule that they transport, closes from that side, opens on the other side, and release the molecule on that side. They go through this cycle again and again to transport more molecules. Because of this lengthy process, the rate of transport through a carrier is slower than through the channels. Now carriers can be of three types: uniporters, symporters, and antiporters. Uniporters move a single type of substance down its electrochemical gradient. For example, glucose transporters move glucose down the concentration gradient. Other example of uniporters is urea transporters. Now symporters and antiporters. They move one type of molecule downhill and utilize the energy from their movement to move other type of molecules uphill. In symporters, both the substances move in the same direction. For example, sodium glucose cotransporter in the apical membrane of proximal tubule. Here, sodium concentration is high in the lumen as compared to inside the cell, and glucose concentration is higher inside as compared to the lumen. This cotransporter couples the movement of glucose with downhill movement of sodium. So glucose is moved uphill. This type of transport is called secondary active transport.